For the scientific research presentation, I did cell theory. And the cell theory was made in 1665 by a scientist named Robert Hooke. For the first part of it, the discovery of cell would not have been possible if it wasn't for the advantages to the microscope inserted in learning more about the microscope world. Scientist Robert Hooke improved this design of existing compound microscope, in which I just promoted 1665. His microscope used three lenses: a stage light, which illuminated enlarged specimens. These advantages all allowed Hooke to see something wondrous. When he placed a piece of cork under the microscope, Hooke detailed his observations of this tiny and previously unseen world in his book. To him, the cell's corked as if it was made of tiny cores, which he named pores, which he came to call cells. As in observing the cork cells, he noted that he thought it looked like a honeycomb. He called them cells because it reminded him of cells in a monstrosity. The performed and pores much like a honeycomb, but that the pores of it were not regular. These pores or cells were indeed the first microscopical pores I ever saw, and perhaps that were ever seen, for I had not met with any writer or person that had made any or mention of them before this. Not long after Hooke's discovery, Dutch scientist Leeuwenhoek detected other hidden minisexual organisms, bacteria, and protozoa. It was unsurprising that Leeuwenhoek would make such a discovery. He was a master microscope maker and perfected design of the simple microscope, which only had single lens, enabling it to magnify any and or object by around 200 to 300 times its original size. What Van saw with these microscopes was bacteria and protozoa, but he called these tiny creatures animalcules. German scientists Theodor Schwann and Matthias Schlegel studied cells of animals and plants respectively. These scientists identified key differences between the two cells and put forth the idea that cells were the fundamental units of both plants and animals. However, Schwann and Schlegel misunderstood how cells grew. Schlegel believed that the cells were seeded by the nucleus and grew from there. Similarly, Schwann claimed that the animal cells crystallized from the material between other cells. Eventually, other scientists began to uncover the truth. Another piece of cell theory puzzle was identified by Rudolf Virchow in 1855, who stated that the cells are generally generated by existing cells. Virchow later stated that by generating, he means like the cells splitting from like one being one and it splitting in two, and eventually two make four, then 4 makes 8, then 8 makes 16, and so on. Well, at the turn of the century, attention began to shift toward cytogenesis, whatever you want to say, which aimed to link the study of cells. The discovery of cells continued to impact science 100 years later with the discovery of stem cells, the undifferentiated cells that have yet to develop into more specified cells. Scientists began devouring embryonic stem cells from mice in the 1980s and 1998. James Thompson isolated human embryonic stem cells and developed cell lines. The discovery of cells had a greater impact on science than Hooke could have ever dreamed in 1665. In addition to giving us a fundamental understanding of the building blocks of all living organisms and non-living, well, not, I messed up there. The discovery of the cell has led to advances in medical technology and treatment. Today, scientists are working on personalization medicine, which would allow us to grow stem cells from our very own cells and then use them to understand disease processes. All of this and more grew from a single observation of the cell in a thorax, created from Hooke's invention in 1665 that led from a 3-glass microscope. And that's all I got for you.